ordinary human T cells. It is regulatory friendly by offering full traceability and drug master file. These benefits were shown in a recent peer-reviewed publication in July. Wanted to test CPS immune cells in an in vivo model of immunotherapy. We teamed up with our collaborators at the Riley Laboratory at the University of Pennsylvania to evaluate the performance of CAR-19 cells grown in CPS immune cells versus those grown in human AD serum. They have an established in vivo model of leukemia where they are able to measure tumor burden over time, efficacy of treatment, and survival of the mice groups. Also, prior to the in vivo study, they compared the expansion, transduction, and phenotype of the cells in both conditions. Data from 3 independent experiments showed comparable expansion of CAR-19 cells in vitro when grown in human AD serum or immune cells are. These cells also showed comparable transduction efficiencies as shown in the bottom right of the slide. Phenotype of the T cells was also comparable, showing similar expression patterns for markers such as CD62L, CD27, and CCR7. Cytotoxic capacity of the T cells was also similar when compared to controls in an 18-hour cytotoxicity assay. After establishing that the cells behave the same when grown in human serum supplemented medium versus immune cell supplemented medium, the cells were cryopreserved. We proceeded to use the U-Penn leukemia model in immunodeficient NSG mice to determine differences in efficacy between CAR-19 cells grown in medium supplemented with immune cell versus human AD serum. NSG mice were injected with 1 million luciferase expressing non-6 leukemia cells. After establishment of leukemia for 7 days, mice were imaged and randomized for treatment with the indicated T cell populations at 10 million T cells per mouse. We used untransduced T cells as a negative control. As you see on the left panel, the day-1 figure represents tumor burden before T cell infusion. And at day 13, CAR T cells were able to control leukemia after infusion, while the negative control group of mice succumbed to leukemia. What's interesting and notable is that immune cells grown CAR T cells control leukemia in 5 out of 6 mice by day 49, while serum-grown CAR T cells failed to control leukemia by the same time. Difference in potency was correlated with a better engraftment of the infused cells grown in immune cells and longer persistence in vivo when compared to cells grown in human AD serum, as shown on the bottom panel. Differences in efficacy translated to a better survival of the mouse group treated with CAR-19 cells grown in immune cells as shown here in the survival plot. In conclusion, this data shows that T cell expansion in CPS immune cells result in similar T cell yields, phenotype, and effector function in vitro when compared to T cells grown in medium supplemented with human AD serum. Interestingly, in vivo, we saw differences in engraftment that correlated with efficacy. Namely, T cells grown in immune cells engrafted better and persisted longer than T cells grown in medium supplemented with human AD serum. This improved engraftment and persistence correlated with a more durable tumor control. A big thank you goes to Jim Riley and his team at the Translational Research Program at the University of Pennsylvania for performing the work on CAR-19 that I showed today as a case study. Thank you for watching. Goodbye.
Thank you for joining us for today's presentation, in which we will be reviewing the pluripotent stem cell culture systems provided by Thermo Fisher Scientific. This presentation was put together to assist you in identifying the culture system which best fits your workflow needs, as well as to provide you with tips for successful culture in each of the systems. The pluripotent stem cell culture system broadly encompasses your cells, such as your ESCs or iPSCs, the medium and extracellular matrix in which your cells are cultured, as well as the passaging reagents which are used in routine culture and expansion of your PSCs. Additionally, cryopreservation reagents play an important role in ensuring cell line security upon expansion. In this presentation, we will be reviewing the tips and tricks for successful pluripotent stem cell culture in the knockout serum replacement feeder-dependent system, as well as the StemPro HESCS-FM and essentially feeder-free systems. Additional information will also be provided regarding transition of existing pluripotent stem cell lines from feeder-dependent to feeder-free culture systems, as well as cryopreservation reagents to ensure cell line security during expansion of your derived pluripotent stem cells. Here we highlight the progression of pluripotent stem cell culture systems over time. Initial PSC cultures utilized the richness of fetal bovine serum to support the health of pluripotent stem cells cultured on feeders, yet there was a large amount of inconsistency between SBS blocks. In 1997, in a move to eliminate the inconsistency of using fetal bovine serum, knockout serum replacement was launched. Since that time, thousands of papers have been published, and this well-established system is still used today. To further simplify the system and eliminate the need for feeder cells, researchers then moved to mouse embryonic fibroblasts or human fibroblast conditioned medium to support culture of their cells. Further refinements provided for defined feeder-free growth medium containing bovine serum albumin, yet there were still lots of inconsistencies noted with the bovine serum albumin component. Finally, Essential 8 was developed by Gokao Chen and colleagues in James Thompson's lab. This system provides a xeno-free medium and matrix for feeder-free culture of PSCs. Here, the system reduced the variability compared with existing feeder-free culture media and provided a cost-effective and robust system for culture of PSCs. In the following slides, we will summarize the feeder-dependent and feeder-free culture systems provided by Thermo Fisher Scientific, as well as the best practices for culture in each system. As mentioned earlier, knockout serum replacement, or KSR, is used for robust feeder-dependent culture of PSCs. This system is provided as a kit comprised of a basal medium, the knockout serum replacement, as well as a basic FDF to support the maintenance of pluripotency of your stem cells. Additional components which are not included in the kit that are added to the complete medium include MEM non-essential amino acids as well as beta-mercaptoethanol. As this is a feeder-dependent culture system, PSCs are grown in colonies on a layer of irradiated meth, which are seeded onto attachment vector. In the image on the bottom left of your screen, you can see the iPSCs growing on a layer of mouse embryonic fibroblasts. There are two commonly used methods for passaging your cells, either manual passaging or enzymatic. In manual passaging, typically syringe needles or a StemPro Easy Roller Passage tool are used. Alternatively, for enzymatic passaging, collagenase or disbase 2 are used. Examples of these passaging methods will be reviewed on subsequent slides. Looking at the workflow for KSR-based medium culture, plates are initially coated with attachment vector onto which the meths are seeded. The pluripotent stem cells are then seeded onto this meth layer in the presence of KSR-based medium. And over time, routine manual cleanup of the cultures is carried out to remove areas of aberrant differentiation. And finally, the cells are passaged. Here we will cover some considerations for the systems to help you ensure successful culture of your PSC. For matrix considerations, the feeder layer may be prepared